Robin. I really appreciate it. So let me pull up the presentation from the beginning. Right, and if everyone doesn't mind just muting their mic, give me one sec. We should be good to go. So Dermomycoides is an emerging pathogen, and as a result, its taxonomy and origin is still unknown. So in the literature, it's referred to under a wide variety of names, including Purkinjean clade organism, Purkinjea-like organism, and Dermomycoides, which is what I'll be using throughout the remainder of my thesis. It's a protist parasite whose infectious stage is characterized as spherical zoospores with a single flagellum. And if you look in this picture to the top right, you could see these spherical zoospores within the intestine of a green frog. Now, this pathogen uses a single host life cycle, but can have additional transport hosts that help expand its range. And the known single host that the pathogen reproduces asexually in has been documented in a wide variety of amphibian hosts, and especially in their larvae. Now, gross characteristics, or characteristics you could see with the naked eye of an infected individual, include hemorrhaging of the liver and spleen, observed as small red or black dots, like what you see in the arrows to the bottom right. And zoospores of this pathogen can be found in nearly all of the tissues of an infected individual, but are most predominant in the liver and spleen. The known locations for this pathogen have been documented across the United States, even as far-reaching as Alaska. But when we look at this map, what you see is the vast majority of mass mortality events associated with this disease, documented by small circles, has been in the east and southeast. And at least one of these mass mortality events occurred within the range of the Dusky Gopher Frog. Originally ranging from Alabama all the way over into Louisiana, the Dusky Gopher Frog is currently only found in 4 counties of southern Mississippi. Originally listed in 2001, only approximately 200 breeding adults in the entire Glens Pond metapopulation. And due to head starting captivarium programs for the recovery of the species, we now have 4 artificially propagated populations as well, but we don't have a clear estimate on the number of animals within each of those populations. But because the Dusky Gopher Frog male takes 2 to 3 years to become sexually mature, and the female takes 3 to 5 years to become sexually mature, years of low or no recruitment of juveniles into these populations causes long-term delays in the overall species recovery. And these years of low recruitment can be caused either by a shortened hydro period, where the pond dries before tadpoles are able to finish metamorphosis, or a mass mortality event caused by infection of Dermomycoides. And when we look at the life cycles of these 2 species, what we see is that they're directly intertwined. As adult gopher frogs migrate into the pond to breed in the late winter and early spring, they'll deposit their eggs within the pond basin. These eggs then develop into tadpoles, which undergo metamorphosis in the mid to late summer. And while these tadpoles are in the pond basin undergoing development and metamorphosis, they're being exposed to infectious zoospores of Dermomycoides. Not only that, but they're also being exposed to non-infectious spores that are present in the water column. But between a pH of 5.5 and 7, these non-infectious spores are able to hatch into infectious zoospores, which can penetrate and directly infect the tadpoles. Now, to date, no mortality event has been documented as 100% mortality of all individuals, but they have been consistently over 90% mortality of potentially infected gopher frogs for these breeding seasons that experience mass mortality events. And so understanding what's allowing a very small number of tadpoles to survive to metamorphosis, while the vast majority of them experience mortality associated with this pathogen, has become an increased concern for conservation efforts and the long-term recovery of the Dusky Gopher Frog population. So my thesis was aimed at understanding how zoospore level and tadpole developmental stage on mortality affects marine tadpoles exposed to Dermomycoides, and also understanding acquired resistance to Dermomycoides in Dusky Gopher Frog tadpoles specifically. Another way to think about this is I wanted to first understand what's causing mortality in these tadpoles, and then understand what's allowing some to survive to metamorphosis, and if that could be the result of acquired resistance. In order to answer these objectives, I collected wild eggs deposited in the Glens Pond Basin within 24 hours of their reproduction. At the time of collection, I gathered an additional 3 eggs that was analyzed using quantitative PCR so that I would know the baseline infection level of Dermomycoides before any experimental exposure. The entire collected egg clutch was taken to the Harrison Experimental Forest, where it was rinsed with ultrafiltered water to remove any excess contaminants before being transported to the Gulf Coast Research Lab, where they are maintained in either the wet lab or maturation lab among the Halstead campus. In order to get the material for my artificial infections, I extracted the liver and intestine from dead infected Dusky Gopher Frog tadpoles gathered either at the Harrison Experimental Forest or the Mississippi Sandhill Crane National Wildlife Refuge, because these clutches came from the same genetic background. I mashed the liver and intestine into a homologous slurry that was filtered through 0.5 micron filter paper before being desiccated for 1 hour and then resuspended in 6.5 parts per thousand sodium chloride in order to maximize the hatching rate of spores into zoospores. This zoospore dose was calculated using a hemocytometer, where I would count the number of individual spores within the center square of the reader. And as you can see in the picture to the very bottom, there's a single spore within this square. For analyzing the infection loads of individuals collected due to mortality or at the endpoint of each of my experiments, I used quantitative PCR, also known as qPCR. This is a method for detecting, characterizing, and quantifying the amount of nucleic acids present within a sample. Just like regular PCR, the DNA from your sample is denatured, alleled, and elongated using temperature or heat over a period of time. And these cycles of temperature and heat fluctuations are what's called an amplification cycle. However, unlike regular PCR, qPCR uses a binding dye that creates fluorescent labels that can be measured with a fluorescitometer with each cycle. And it does so by binding a probe that's specific to the DNA sequence that you want. So only that sequence is amplified and measured. And because it's specific to that sequence, which gets amplified over each cycle, the signal is increased with increasing amounts of the replicated DNA that you're looking for specifically. For my project, I used the TACMAN probe that you see in the bottom right, following the procedure of Karawacki et al. Because this probe is specific to Dermomycoides and has been tested to quantify the amount of Dermomycoides DNA present across a wide variety of amphibian DNA, I followed this procedure in order to understand the infection levels of my experimental exposures from baseline to endpoint. For my analysis, I used a linear mixed model to analyze average days to mortality and infection level with the package Limer in RStudio. For survival, I analyzed SurviveFit and survival also in RStudio. And when appropriate, I used a Tukey's post hoc test. To understand my first objective of investigating the effects of zoospore level and tadpole developmental stage, I needed to understand the terms of susceptibility and sensitivity. Susceptibility is how likely it is for a tadpole to become infected, whereas sensitivity is how severe that infection becomes once already established. This can also be thought of as resistance versus tolerance. Susceptibility fluctuates throughout metamorphosis as alleles known for regulating the immune system are routinely turned on and off throughout the process of organogenesis, or growing organs throughout metamorphosis. And as a result, susceptibility should be dependent on developmental stage. To test this, I conducted an experiment where I looked at the susceptibility of gopher frog tadpoles specifically across different developmental stages. Now, sensitivity is known to be species specific because for the dusky gopher frog, they experienced a reduced population, genetic bottleneck, and inbreeding depression. However, southern leopard frogs, which occupy much of the same range as dusky gopher frogs, have a very similar life history, but are an extremely common species, have a much higher genetic diversity, and should be less sensitive to infection as a result of that. But even in the most tolerant or intolerant species, there is typically a threshold effect where infection can be tolerated in the organism up until this point, at which the tadpole is no longer able to reduce the detrimental effects of the infection, and mortality occurs. So to answer this question, I conducted a second experiment that looked at sensitivity of gopher frogs versus leopard frog tadpoles exposed to different zoospore quantities in order to see if gopher frogs do have a higher sensitivity to Dermomycoides than leopard frogs, which are a far more common and genetically diverse species. For my first experiment, I looked at the work done by Josh Cook and Matt Atkinson, who both exposed southern leopard frogs to 100 zoospores per microliter. However, these two researchers used different metrics for describing the developmental stage or time at which their tadpoles were infected. Josh Cook used hours after deposition into the pond basin, and Matt Atkinson used Gosner staging, which is a standardized method for characterizing development of larval amphibians. And because even tadpoles of the same species, depending on temperature, genetics, and a wide variety of other environmental factors, can develop at different rates, it's not entirely clear if these two developmental staging treatments overlap, leading us to the possibility of a knowledge gap in susceptibility of leopard frogs to Dermomycoides throughout their metamorphosis. But when we look at both Josh Cook's work, which is in this dark blue, and the first 5 bars of the graph versus Matt Atkinson, we see that both researchers saw low survival in the earliest developmental treatments used in their experiments. And so for my thesis and for my experiment, I targeted the developmental stages
We lost the video.
embryos, which can be anywhere between Gosner stages 11 and 19. At this point, the eggs start to elongate. Tadpoles exposed as hatchlings. At this point, they are removed from the egg mass, and between a Gosner stage of 20 and 22. And finally, tadpoles exposed 2 weeks after hatching from the egg mass. At this point, they have obvious mouth parts and are free swimming. And they're also at a Gosner stage of 25, which was the lowest developmental stage used by Matt Atkinson. Because my animals were collected from the wild population, I had 2 control groups, one kept in the same room as the animals being experimentally exposed, and one kept in the separate quarantine room, so that I could look at the differences in infection level between my in-room and out-of-room controls in case there was any accidental contamination, and see how the infection grows over time in animals that definitely were not exposed to a secondary exposure. For my experimental design, I artificially exposed tadpoles in groups of 10 to each developmental stage treatment. And each treatment consisted of 3 replicates that represented a different egg mass, as you could see here in my tank map below. When the tadpoles developed to the stage needed for their treatment group, I exposed them in small plastic dishes for 30 minutes before transferring them to 2.5-liter aquaria, where they were maintained for the remainder of the experiment. 2 weeks after that group's initial exposure, I collected one individual from each replicate treatment, and 4 weeks after the initial exposure, I collected all surviving animals for the endpoint of the experiment. When I analyzed average days to mortality, I found that there was a significant decrease in days to mortality with increasing developmental stage. And that right after tadpoles develop from eggs to embryos, there's a slight decrease in days to mortality, which is significant. But as soon as they hatch from the egg mass, they die much faster and are significantly lower 2 weeks after hatching, when they're not even mildly attached to the egg mass anymore. And so that tells me that tadpoles exposed as hatchlings or 2 weeks post-hatch were at their most susceptible. But when looking at sensitivity of the gopher frogs, you'll notice that I used 50 zoospores per microliter in that experiment, whereas Josh and Matt used 100 zoospores per microliter with leopard frogs. And I did this primarily because Josh and Matt were the only other researchers who had ever done experimental exposures using Dermomycoides. And they used 100 zoospores per milliliter as an arbitrary amount and then kept it because it was the only thing in the literature. So in 2017, I conducted a preliminary study that exposed gopher frog tadpoles at Gosner stage 25 to 0, 25, 49, 74, 166, or 250 zoospores per microliter, and followed them for 107 days, which is the average days to metamorphosis in head-started individuals at the Harrison Experimental Forest. And I found no significant difference in average days to mortality across any of the treatments. And so this meant that my tadpoles who were exposed to 0 zoospores, or nothing above their baseline contamination level, didn't die any faster than tadpoles exposed to 250 zoospores per microliter. So under the guidance of Dr. Overstreet in 2019, I conducted a follow-up study after we decided that zoospore range was likely not dramatic enough to elicit any differences in mortality over time. And in this follow-up study, I used both gopher frogs and leopard frog tadpoles exposed to 0, 250, 500, or 750 zoospores per microliter. And again, I used both species in order to see if there is any difference in sensitivity to infection of Dermomycoides among the species. Now, I used 5 tadpoles per replicate treatment in order to maximize the number of replicates for each of my treatments. And as a result, I had 11 gopher frog replicates and 3 leopard frog replicates that were maintained in 1.5-liter plastic containers and fed 1 algal wafer a week. At Gosner stage 25, they were exposed to their treatment groups for 30 minutes before transferred to the containers. And 2 weeks after this exposure infection, a single individual was collected from each replicate treatment, and 4 weeks after the initial infection, all surviving individuals were collected for the endpoint of the experiment. When I analyzed average days to mortality again, I once again saw no significant difference in average days to mortality between my control group and my highest dose treatment group, which was 750 zoospores per microliter. However, in my out-of-room control group, which had no chance of cross-contamination and only had that baseline infection level, what I saw is that leopard frogs had a higher days to mortality or survived longer than dusky gopher frogs, which is an interesting bit to note. So what we now know is that developmental stage at time of initial infection does play a role in mortality rate, and tadpoles are at their most susceptible right after hatching from the egg mass. And while no zoospore density threshold is found for either gopher frog or leopard frog tadpoles, we do now know that the two species are equally sensitive to infection of Dermomycoides. So going back to the work done by Josh Cook and Matt Atkinson and mapping their average survival that they observed among their southern leopard frog tadpoles exposed to 100 zoospores per microliter, and pairing that with the survival I observed with gopher frogs exposed to 50 zoospores per microliter, what we see is a clear fluctuation in susceptibility throughout metamorphosis. Now, this is likely due to the fact that the immune system is known to fluctuate throughout metamorphosis because organogenesis, or growing organs, is a highly metabolically costly endeavor, as is immune system functioning. And so at hatchlings and 2 weeks post-hatch is a known time of rapid growth of organs and developmental features, possibly leading to this heightened susceptibility. And the very low mortality observed in our egg masses is likely due to the fact that the jelly layer surrounding the yolk is serving as a protective barrier against zoospores and other contaminants. But as soon as those eggs began to elongate into embryos, that jelly layer was compromised and the animals became more susceptible to infection. I didn't find any significant difference in either days to mortality or survival in the preliminary or follow-up study, which means that I don't know if there is a tolerance threshold to Dermomycoides for either dusky gopher frogs or southern leopard frogs. But we do now know that southern leopard frogs are as sensitive to infection of Dermomycoides as gopher frogs, and as a result, they could be used as an excellent proxy species for future studies on this pathogen and in making management recommendations based off of that work for the dusky gopher frog, as these animals do share the same territorial range, life history, and sensitivity to infection of Dermomycoides. Now, going back to our research objectives, we now understand that susceptibility to Dermomycoides fluctuates throughout metamorphosis, and that tadpoles are at their most susceptible after hatching. And we also know that zoospore exposure level isn't necessarily correlated with mortality associated with Dermomycoides. And taking the results of those 2 experiments and my first objective, I approached my second objective, which was understanding acquired resistance to Dermomycoides in dusky gopher frog tadpoles. Now, I once again had to understand the terms of resistance and tolerance. Resistance is the ability of a host to avoid or decrease infection, whereas tolerance is the ability of a host to mitigate the detrimental effects of infection. Acquired resistance is the ability of a host to change behavior, activate an immune response, or reduce infection loads upon multiple exposures in an individual lifetime. And as a result, it's a combination of both resistance and tolerance strategies. To illustrate this, imagine the blue circle is the Glens Pond population, and all of the tadpoles that you see within it are all of the tadpoles that can survive to the metamorphosis, resulting in the juvenile frogs surrounding the pond basin. And all of these juvenile frogs are going to survive to the first reproduction and can be counted towards the population for that year. Now, because dusky gopher frogs breed in the late winter and early spring, these breeding events are associated with periods of high rainfall, which increases the water level of the pond basin as it's filled and dried entirely based off of rainwater. Now, as the water level rises, so does the pH. And we know, based off of the work of Josh Cook, that between a pH of 5.5 and 7, non-infectious spores of Dermomycoides are able to hatch into infectious spores that can penetrate and directly infect tadpoles. So in these large rain events, these tadpoles are likely exposed to zoospores of Dermomycoides, which can cause a mass mortality event, resulting in a loss in the potential number of juveniles for that breeding season. Now, even in a year where the water level stabilizes, the pH is reduced, and the number of zoospores goes down, you will always lose a portion of your population due to predation. But because the larval period can be anywhere between the late winter and to late summer for these animals, it's likely that they experience multiple high rain events that once again increase the water level, increasing the pH and exposing them to zoospores once again. However, if tadpoles are able to acquire resistance after surviving that initial exposure to Dermomycoides, then instead of seeing mortality in the remaining 3 tadpoles within our pond basin, only 1 may experience mortality, whereas the rest survive, giving us recruitment of 2 of our potential 7 juveniles. To approach this in a laboratory setting, I conducted a split plot design where I exposed tadpoles to an initial high, low, or 0 dose of zoospores, where my high dose was 250 zoospores per microliter, low was 50, and 0 was 0. 2 weeks after the initial infection, I collected 5 animals from each replicate treatment and exposed the remaining to a second high, low, or 0 dose of zoospores of Dermomycoides. All surviving animals were collected 4 weeks after the initial exposure. And when looking at average days to mortality, I once again saw no significant difference among treatments either before the secondary challenge dose or after the secondary challenge dose, or even in tadpoles that received no secondary challenge dose. But out of curiosity, I plotted the average days to mortality of initial and second exposure doses together, where the initial dose is classified by different lines, where our low dose is this dark red line that has no marker. High dose is a yellow line with a diamond. 0 is a blue line with a triangle, and my out of room control is this green box over here, as it was never exposed to anything. And along the x-axis, we see the second exposure dose group. So all of these animals, for example, had no challenge dose, a low challenge dose, or a high challenge dose. And what we see is that across all tadpoles that had that low initial zoospore dose group, they had the lowest days to mortality across all second challenge exposure groups. And when looking at average survival to endpoint across the initial treatments, what we see is that before that challenge dose is administered, tadpoles that have that low initial dose had the lowest days to mortality, had the lowest survival. So looking at the endpoint and the combination of all of the treatments together, challenge and initial, what I found is that tadpoles that received a low initial and high challenge dose of zoospores had the low, and tadpoles that received a low initial and 0 challenge dose of zoospores had the lowest survival to endpoint. Where if you look, the tadpoles that received a low initial treatment and 0 challenge only had a 0% survival to endpoint, whereas those that received a low initial and high challenge had only a 3% survival to endpoint, which means that those combinations were the absolute worst out of all of the treatments for survival. When looking at average infection level after analysis using qPCR, I found no significant difference in infection levels of any of my individuals and any of my treatments to the endpoint of the experiment. But just looking at the individuals that only received an initial treatment and not a challenge dose treatment, I was able to analyze the growth of this pathogen from midpoint to endpoint of the experiment. And what you see is that there's an overall average increase in infection level over time. But this infection varies among egg masses. So if we look at egg mass 11, the growth of the infection responds as one would expect, where in our 0 and low treatment group, the growth is very low or basically none. But as soon as they receive that high initial dose group, the infection increased over time. But for tadpoles from egg mass 13, in the 0 dose group, they grew over time slightly, but when exposed to either the low or high initial treatment, they actually were able to decrease their infection over time, which is really interesting. And it tells us that infection is really dependent on the egg mass from which these tadpoles originate. But across all treatments, we did see the strategies of resistance and tolerance used, where tadpoles within our control group showed and displayed natural tolerance levels because we know that they came into the experiment already infected with a low baseline level of Dermomycoides. And tadpoles within our high exposure group demonstrated resistance in that this high exposure of 250 zoospores per microliter may have been high enough to trigger an immune response. But the high mortality in our tadpoles exposed to a low initial dose of zoospores of Dermomycoides may have resulted in the fact that this dose was both too low to trigger an immune response and also too high for a natural tolerance of the tadpole, resulting in the high mortality. So going back to our research objectives, where we wanted to understand the effects of zoospore level and tadpole developmental stage on mortality of brained tadpoles, we now know that susceptibility fluctuates throughout metamorphosis and that dusky gopher frogs are at their most vulnerable right after hatching from the egg mass. And for understanding acquired resistance to Dermomycoides and dusky gopher frog tadpoles, we know that infection can be tolerated and that the tadpoles that survive this initial infection are able to acquire resistance to later exposure, but that if they're exposed to very low doses, they're less able to resist a later exposure and more likely to experience mortality. Now, this is important because the acting hypothesis of managers for the past 5 years has been that these periods of high mortality observed in the wild populations is the result of a single high-intensity brain event that increases the amount of zoospores present within the water column, drastically causing a high exposure event, rather than a low-level exposure over a prolonged period of time, which is what the results of my thesis would indicate. For example, in 2018, 86 gopher frog egg masses were documented in the Glens Pond Basin. Now, the average gopher frog egg mass contains 1,200 eggs per mass, but of all of those potential, only 2 gopher frog tadpoles survived their metamorphosis. Because the Glens Pond Basin is monitored once a week for water level, temperature, and pH, we know that over this larval period, the pH of the pond fluctuated between 4.42 and 6. And going back to the hatching rate of spores into infectious zoospores of Dermomycoides, we know that that's at its highest between 5.5 and 7. So for a prolonged period of time, the water column was at the lower range of hatching rate for spores into infectious zoospores, meaning that it's very likely these tadpoles were exposed to a low level of infection over a prolonged period of time, which caused the mass mortality for that overall breeding season. Now, because we saw that high variation in sensitivity and susceptibility to Dermomycoides across different egg masses, we know we can advise head starting and captive rearing programs and managers to really focus on increasing and preserving the genetic diversity of dusky gopher frogs as they continue their repatriation and propagation programs. because in the past 10 years, there have been an increase in the number of restored ponds, and as you can see in the pictures too on the bottom, these are all due to head starting and captive rearing efforts. And so as the number of ponds that managers monitor increases, we need to keep in mind that we need to preserve the genetic diversity of the dusky gopher frog. For example, if your permit restricts you to only collecting 1,000 eggs for an entire breeding season, and you have 3 ponds that among them have 10 egg masses, instead of collecting 300 eggs arbitrarily from each of the ponds, managers need to really be targeting collecting 100 eggs from each of the 10 masses in order to maximize the amount of available genetic diversity that's being represented in their head starting program. Future research with Dermomycoides should investigate the abiotic factors of these breeding ponds that may contribute to Dermomycoides persistence, including how prescribed fire affects spores' ability to hatch into infective zoospores, and if there's a difference between permanent and ephemeral ponds which dry seasonally. Now, because the management of the dusky gopher frog has been wonderfully successful, it's also benefited a wide variety of amphibians that also use these ponds for breeding and reproduction. And we know that Dermomycoides is able to use a wide variety of amphibians for their hosts. So understanding how the disease disperses among infected and uninfected individuals kept together is a crucial next step to understanding mortality rates observed in these pond basins. And finally, thanks to the wonderful work of the Savage Lab at University of Central Florida, we now know that adult gopher frogs, Rana capita, are able to test positive for Dermomycoides. So it needs to be investigated if these low-level infections that we observed in eggs collected from the wild are actually from being deposited into the pond basin, or if they're being transferred from parent to child during fertilization and amplexus. With that, I would like to thank Robin and Joe Peschman for taking me on as a student and doing this crazy subcontract with me. It has been a really wonderful opportunity, and it's wild to be here today. I'd also like to thank Joe Griffin and Zach Darnell for helping me defend my master's thesis in the middle of a global pandemic. I know that it's been a stressful time for everyone, so I really appreciate this. Janet and Jean for teaching me how to use all the lab equipment and giving me all of the information I need. Matt Atkinson and the Savage Lab for the wonderful exchange of information and ideas. It's been a great collaboration, and I'm excited to do future projects with you guys again. U.S. Fish and Wildlife for funding, and the Mississippi State Natural Science Museum for support, as well as U.S. Fish and Wildlife for support. Alvin, Joyce, and Marianne for giving me all of the resources I needed, even at a very short time crunch. My wonderful friends and family who kept me sane and told me to keep going. I really love you guys. And then finally, I wanted to take a moment to remember Kathy Shelton, who was our contact biologist for the state of Mississippi. And without her, none of this work would be possible. She was a great advocate for the dusky gopher frog and early researchers, and she will be missed. And with that, I am happy to take any questions, and I'm gonna turn off screen sharing for a hot sec so I can see your faces.
Questions?
I do have a question. Sorry,
Yes.
Can you hear me? First,
Yes.
Great job. Second, for the first experiment, you chose to do a second experiment with a higher set of doses. Why did you choose to go higher than lower?
So I chose to go higher because I couldn't go lower. So for that preliminary, you're talking about the different zoospore doses, right?
Right, like I'm saying, why not go to doses like below 10 and see if there was something in there? Maybe you're above the threshold already.
Yeah, so my lowest dose, like my doses between 0 and 25 were
Yeah.
pretty much as low as you can go using a hemocytometer. Yeah. Yeah, that was pretty much as low as I could go.
Okay. Just curious.
Yeah. Well, and the fact that like I didn't see any, there was really, it was pretty much the same from like 0 to 166. It was only at 250 where I started to see a drop, which is what indicated that there might be a trend that only just at 250 we were starting to see that difference.
Okay, thank you.
Yeah. Anybody else?
I have a question. I'm just wondering, like, moving forward, what do you hope this thesis will accomplish in terms of like extending your field and the knowledge of this particular frog and this particular issue further? Like, what are you hoping this contributes to the environment on a larger scale?
That is an excellent question. That is a really, really excellent question. So, like I was saying, our working hypothesis was that these mass mortality events were the result of a single high exposure event. So at least on the management aspect, we had been targeting trying to find these single mass mortality events right after large rainstorms or when the pH spiked, and weren't really monitoring the pond when it was at that lower level. And so we had been missing all of these mortality events. And I contributed to the fact that in southern Mississippi, due to the temperature and relative humidity, unless you're out at the pond the day of or day after, you're not going to see anything because these tadpoles just degrade and disintegrate. But because we now know that mortality is likely more associated with low levels of infection right after the tadpoles hatch from their egg masses, we can target dip net surveys and monitoring efforts to those specific time windows, which is about like a month and is very doable, which gives us a better idea of what to expect in that breeding year, and also when we should be collecting other environmental information such as zoospore counts at the soil level. So it gives us a way better way of monitoring and tracking disease because we now know this baseline information. Does that make sense? It also gives us some hope that, like, if they're able to survive that initial infection, we won't have complete mortality. And that in turn sets us up for future work where if the individuals who were able to acquire resistance and develop through metamorphosis, if they're able to maintain that resistance through adulthood, because we know that they test positive for Dermomycoides thanks to the work of Matt Atkinson and the Savage Lab. So are those adults resistant, or is it just kind of like when you get the flu and the flu virus just lives in your liver forever and waits till you're weak? It's yeah.
Awesome, thank you.
Yeah, no problem. Great question. Really
Hey, Jamie.
great question. I love it. Yes.
Jamie, it's Bones. I have two short questions. Do we know if the spores is killed after a burn? I guess what I'm getting at is like, the first breeding event after a good burn through the whole pond basin, do you see less mortality because there's less dermo? And then the second part was, is there ways to alter the pH to suboptimal levels to have any positive effects?
So great questions. The first one, I don't know, but it would be really pretty straightforward to test that out. You would just need, you could create artificial mesocosms using sediment from a breeding pond, light it on, try and hatch out the number of spores within that sediment, within a portion of that sediment, I apologize. Then light it on fire and then see if you're able to hatch out the same number of spores from a different sample, which would be really cool. And also another great advocate for prescribed fire, which I am always for, but we don't know that officially yet. So that is a study that needs to be done, and it would be really cool. For the second question, sorry, I rambled on about fire for too long. What was your second question?
I was just talking about artificial alteration of pH to like a suboptimal
Yeah.
level.
Yeah, so you could artificially alter the pH of the pond basin, and that was done in, well, it was inadvertently done in about 2006 when the Glens Pond Basin was drying prematurely. So they actually airlifted in giant containers of water to fill the pond basin, but that actually changed the pH and caused a mass mortality because the tadpoles are really sensitive to the pH. So for instance, and each pond is slightly different. So for instance, Glens Pond usually fluctuates between 4 and 5, whereas Pony Ranch will be around like 4.5 to 5.5. But it's usually pretty acidic naturally. And if you were to tamper with that and like artificially lower the pH, you might end up lowering it too much, because not only does the pH affect the zoospores, but it also affects the physiology of the tadpoles, the vegetation. So I personally wouldn't recommend artificially tampering with the pH, just because you would be setting yourself up for mortality associated with all of the other things. Bye, buddy. Thanks for being here.
Thank
That
you.
would also be affected by lower pH.
Thank you.
Sorry, I have one more question too.
Oh, go for it. Absolutely.
Yeah. So you mentioned how narrow the genetic diversity has gotten at this point because of all the die-offs and everything, and how they need to be really careful with choosing which eggs to pick. Do you guys mix eggs between ponds to help kind of inflate that diversity? Or?
Yeah, so that actually has happened, and it's really pretty cool. Hey Joe, what was the last year that Mike's Pond existed? It's like 2005.
The last year that
Mike's
maybe,
Pond.
I don't remember exactly, but it was a while ago. It was
So
at least 10 years
many,
ago.
yeah. So about 10 years ago, there were two different breeding populations and established ponds for the Dusky Gopher Frog, which was Glens Pond and Mike's Pond. Now, the population of Mike's Pond was declining rapidly, and John Tupy, my predecessor, he was able to collect one of the few remaining males of the Mike's Pond population and bring them to Glens Pond, which preserved that genetic diversity and allowed for mixing. But that's really been the only opportunity. I know a bunch of our zoo partners, they really look at the like siblingship and genetic diversity when they do in vitro fertilization, which is really great. For head starting programs at the Mississippi Sandhill Crane National Wildlife Refuge, the Nature Conservancy, and our group, I take a portion of each egg mass that's deposited in the Glens Pond Basin. And then because the Ash South Pond has only just started developing egg masses within the past 2 years, probably next year I'll take a portion of those individuals as well. But that's how we're able to preserve that genetic diversity.
Okay, yeah, thank you.
Yeah, absolutely. Anyone else? No? Last chance.
Any more
Okay.
questions?
Well, thank you all for being here. I really appreciate it. I'm so glad I got to share my work with all of you. I really, really appreciate you all being here for me and my
We're so
research.
proud.
So, thanks. If you don't mind signing off, so only my committee's left, I'm gonna answer a few of their questions, but Thank you again for being here, and I'll be posting the results later.